Hi from the SpaceJam team. SpaceJam enables comprehensive characterization of GeoMx data, GeoMx spatial data, by integrating it with single-cell RNA-seq as well as applying a range of published tools such as CountCorrect or SelfOnDB, as well as customly developed tools for comparing multiple GeoMx sections. And those tools, together with the data, are used to essentially provide functional annotation of disease genes, assess the change in cell composition in this disease state, as well as predict cell-to-cell communication. We put all of this information together into a data portal, which makes it much easier to explore the complexity of data produced by these methods. I want to go a bit more in depth on what exactly we changed in a recently developed model called cell-to-location. This model uses reference signatures of cell types, reference signatures of cell types G, to from the single-cell RNA-seq data to essentially establish special cell type abundance. The model is quite complex and accounts for a number of technical factors such as technology and platform sensitivity from GeoMx to single-cell RNA-seq data. But what we introduced to the previously published version, what we extend compared to the previously published version, is these two essential components, which provide per slide normalization of hybridization efficiencies, as well as enable probe hybridization variation, gene-specific probe hybridization variation. We also use experiment-specific background as well as negative probe binding information. We use this complex model as well as other tools that you just saw before to essentially examine how cell composition changes with age. And I want to highlight a few key results. So specifically, we see that in agreement with the literature, we see an increased abundance of B cells in the tertiary lymphoid aggregates in disease states. We also see a well-known reduction in the podocyte cell abundance. And in addition to this, we discover infiltration of classical and non-classical monocyte-derived macrophages into the glomeruli. And nicely, this corresponds to the functional annotation where the disease glomeruli express many macrophage and granulocyte chemotaxis molecules, as well as other functionally relevant gene sets. On top of this, we use GeoMx data we use differential expression as well as estimated cell type abundance to essentially prioritize cell communication from this data type. And finally, we provide all of that in easy-to-use data portal. And at this point, I want to acknowledge our fantastic team, as well as a few other people who contributed in helpful discussions.